Okay, so let me see here. Presentation mode. Okay, I'll just do a brief set of notes here in description. I just gotta check. Do I have access? Yeah. Okay. So let's go through this. This is just the basics on DNA for today, and then we'll get into genetics next day. So getting to know what DNA, you already have a basic understanding, so I'll go through just a little bit more in depth here from what we studied in grade 9. So first of all, DNA was first isolated by a Swiss biochemist named Johann Friedrich Miescher in 1868, who called them nucleic acids. And after 80 years passed, scientists understood that DNA contained information for carrying out all sorts of different activities in the cell. Okay, so the DNA we found out eventually produced proteins. The DNA acts like a blueprint from which proteins and enzymes are made, and from those proteins and enzymes, all the cell activities are carried out. That includes the building up of new cells, the repair of cells, information that passes in between cells. So all of those functions. So we'll just write down cell activities. Okay. So all of which happens when the DNA is in the nucleus of the cell. So what happens is the DNA becomes unravels, and then copies of it are made, and those copies are sent outside the nucleus, and then from there the proteins and enzymes can be made. Okay. Okay, so we didn't know that though. It goes on here to say, but how the cell did it, how the DNA in the cell did it, was unknown. So they had to determine the structure of the DNA first. And that enabled them to find out how the DNA made copies of itself. So James Watson and Francis Crick were the first to come up with the idea of what the DNA looked like. And they realized that it actually was the form of what we call a double helix. Like a spiral staircase. So if we go down here, we've got a picture of it down here actually. Looks like that. Okay, that's a spiral effect I'm talking about. We call that shape a double helix. You've probably seen stairwells like that are used in places where they're limited with space. So they're called spiral staircases, but the actual shape is called a double helix. Okay, and that you would think that the DNA would be flat like a ladder. Well, it is when it's red, but for the most case or for most situations, it looks like the double helix. The reason why it forms a double helix is because these bonds here. The angle of those bonds causes the shape that we see here. So that's an interesting fact. Let's go back to the top. So why is DNA important? Because the DNA allows genetic information, because genetic information passes from one generation to the next. Genetic info or information passes from one. That is what DNA allows to happen. It's acting like a blueprint and contains the very genes which contains the information that's passed on from generation to generation. So it controls cellular activity, by controlling the production, I mentioned it earlier, of proteins and enzymes. Okay, so the protein is used to build up cells that creates tissue, like muscle tissue, and the enzymes are used to carry out functions in the body. Okay, so what is DNA? DNA, as I said, is located in the nucleus, in the center of the cell, and the molecules are made up of thousands of repeating units called nucleotides. Okay, so what's a nucleotide? A nucleotide is, these are nucleotides here. So that's a nucleotide here. And that's another nucleotide there. And that's another nucleotide here. And that's another one here. So what do they contain? They contain, you're already familiar from grade 8 or 9 that the DNA contains what we call bases. So that's adenine there that I just pointed to. And the nucleotides also contain a phosphorus group and a sugar. Sugars here. Okay. Those three things make up the nucleotide. So each nucleotide has a phosphate group. I've got allergies. Sorry about that. 5 carbon. Deoxyribose sugars. Simply called sugars. And then the bases, the nitrogen bases. Okay, so here are the bases here. There's 4 bases in total, so 1, 2, 3, 4. So the purines are called adenine and guanine. We use A for adenine, and then guanine, we obviously abbreviate with G. Okay, so you can see what are they contain. Oh, excuse me. They contain nitrogen, as we said. They're called nitrogen bases. There's nitrogen in them, and then they're made up of other nitrogen and carbon and oxygen structures. Very closely. I mean, if you look at the structures, they're very similar, right? That's the basic structure is similar. This structure here is identical to this. What's different? This one contains an oxygen and a nitrogen here. Okay. I guess actually that one does too. That contains nitrogen there. So they both contain the NH2. So that they both have that in common. So what's different? The double bonded oxygen here. That's one of the main differences. So there's slight variations between these two. But you can see they're closely related. These two on top are also closely related. Okay. So you got 1, 2, 3, 4, 5, 6. So heptapenta. Pentagrams, right? So you can see that there's pentagram structures in all four. This top pair only has one pentagram structure each. Okay, so the others, again, that are similar that I just pointed out are the thymine, that's this one, and the cytosine. That's this one here. Okay, so let's go on here. So it says because the base pairs together, they pair together in only one way. The two strands of DNA molecules are complementary. So you already know this rule, but I just remind you of it. Adenine always pairs with thymine. Adenine always pairs with thymine. Okay. So A always pairs with T. And guanine always pairs with cytosine. Okay. So what are those? These are what we call again the base pairs. Those bases, the pair of them make up, if you were to look at the DNA, I'm pointing out where they're located in the DNA, they make up the rungs of the ladder, the steps of the ladder. And they always come in certain pairs. T always combines with A, and G always combines with C. It doesn't matter which one comes first. Okay, so you can have TA, you can have AT, both of which means, when it's being deciphered, means completely two different things. You can have GC or CG. So you have essentially a 4-letter alphabet. Okay, so let's go back to this. So each pair, they're held together by hydrogen bonds. So the A and the T are held together by two hydrogen bonds, and the G and the C are held together by 3. So 3 H bonds. As opposed to the one on top, which are 2 H bonds. Okay, now what's the complementary DNA structure? If we look at this here, you know what each of these letters is paired with. A is always paired with T. And C is always paired with G. And that last one should be a G. Excuse me here, I'm going to get some Kleenex. Oh boy. Okay, so let's go on here. So these are some important points. Since adenine must pair with thymine and guanine must pair with cytosine, the amount of A always equals the amount of T, and then the amount of C is always equal to the amount of G. And then we went over this. This is the structure of each nucleotide. It's made up of a sugar, made up of a phosphate group, and it's made up of a base pair, a nitrogen base. That I'm referring to each of these, right? Okay. We'll go down to here. So this is the double helix structure I was referring to. And again, that structure comes about because of the angle of the bonds in the sugar groups. Let's go down here. So the double helix model was developed by Watson and Crick. And the DNA model is made up of two connected chains. Of nucleotides, forming a ladder-like structure. So two connected chains. This is one chain. I mean, it'd be a lot longer than this, but that's the right side. That's one chain. That's another chain. These would be a lot longer. The side of the ladder are made up of phosphate and the sugar. And we can just put deoxyribose. Sugar groups, molecules. Rungs of the ladder are made up of a pair of nitrogen bases bonded by hydrogen bonds. And then the strands are, like I said, very long. Strands are very long. Like I was hinting at here, the strands or the chains are very long, a lot longer than what I pointed out here. So the information contained in the linear sequence of the bases in the polynucleotide chain. Okay, so that's where the information is contained. It's contained in the sequencing. What does that mean? The information is dependent upon how these groups here, what order they come in. In other words, is the T first or is the G first? Or is there a C in front? Or is there an A in front? And what is it followed by? Okay, I do know this, that a group of, this is getting messy here, but a group of all three bases is enough to make up one amino acid, which goes into making proteins. So let's go back here. DNA for all organisms have has the same structure, but different sequences equals different information. Okay, A plus C and G plus C are different for different species. That means the order is different. That's what makes the genes different. Okay, question, why would DNA need to make copies of itself? So DNA can make exact copies of itself. Okay, why does it need to do that? Because it has to obviously make new cells that are clones of the parent. Replication occurs during the process of mitosis. Oh, that should be an and meiosis. Okay, so how does that happen? Well, this is the beginning of the process. This is how DNA makes a copy of itself. First of all, it becomes unraveled. It's originally in this form here, in the form of a double helix, and then it unravels, straightens out flat like a ladder, and then it splits, as it's shown in the second diagram. And when it splits or it unzips in half, new bases are able to join at each of these points. That forms the ladder on the left. And then similarly, use a different color. Bases connect with each of those pink points that I highlighted here to form the ladder on the right. Okay, so each of these ladders is a hybrid of old and new bases. Okay, so we're going to leave it at this point for today, and then I'll go on to some other lessons tomorrow and Wednesday. I hope everyone's doing well, and I'll talk to you soon. You can, again, you can use this lecture in the same way that I did. You can just fill out the notes as I go along accordingly. And you know what? If you want, for further study, you can make up questions as well. And I wish I had more time to get into that in terms of the course, but at least this gives some idea of the foundation of genetics. So that is a review of DNAs. We need to know about DNA in general because that ties heavily into genes and the topic of genetics. Okay, take care. I will talk to you soon. See you tomorrow.